Hello dear student and researcher. In the today lecture we are going to discuss the effect of corona disease COVID-19 on the sperm quality. In the recent study published in the Journal of Medical Virology, researchers examined the effect of corona disease COVID-19 on sperm qualities. Actually, about the study, in the present study, researchers evaluated the impact of COVID-19 on sperm morphology, motility, and count. And males aged 20 to 50 years visiting in the in vitro fertilization unit at the Adena City Training and Research Hospital were included. Male who tested SARS-CoV-2 positive in the past 3 months and those with less than 1.5 ml of semen were excluded. Patients with a confirmed history of SARS-CoV-2 infection in the past 4 months to 1 year were included in the COVID-19 positive group, and those without COVID-19 served as control. There was no information on whether participants underwent a spermogram before COVID-19. Samples were not obtained from those severe COVID-19 patients. Now you know that severe acute respiratory syndrome coronavirus 2 infection typically causes respiratory symptoms and fever, but in some cases COVID-19 may be severe with multi-organ involvement. The host receptor for SARS coronavirus 2 is angiotensin converting enzyme 2 ACE2. Actually, this is widely expressed in the lungs and the spermatogonia, Sertoli cell and Leydig cells. Actually, Sertoli cell and Leydig cell, they are found in the testes, and actually these two cells help in the spermatogenesis, the formation of sperm, and also help in the formation of testosterone. It has been suggested that gonadal function may be affected due to inflammatory responses against SARS coronavirus 2. Moreover, immune responses in the testes negatively affect spermatogenesis. Therefore, more research is required to understand the effects of SARS coronavirus 2 infection on the male reproductive system. In these studies, overall 200 male samples were included in the study, equally split into COVID-19 positive and COVID-19 negative. Of the SARS-CoV-2 patient positive males, 40 had a mild clinical course and 52 developed moderate symptoms. The median age was 30 and 30.5 years for COVID-19 negative and positive participant respectively. The median sexual abstinence period was 4 days for COVID-19 negative male and 3 days for those in the COVID-19 positive group. The sperm concentration of COVID-19 negative participant was significantly higher than COVID-19 positive males. Semen volume and sperm morphology and motility did not significantly differ between the two groups. 4 male in the COVID positive group, but none in the COVID negative had azoosperma. Sperm were enumerated under a phase contrast microscope and were classified as types A, that were rapidly progressively motile, slow type B were progressively slow progressively motile, and type C were motile in situ, and type D were immotile. The proportion of each sperm type and total motility were determined. Sample with a concentration of more than 5 10 raised to the power 6 ml were stained and examined for the Kruger criteria. After the conclusion, the researcher observed significantly elevated sperm concentration in male without COVID-19 infection relative to those who had been COVID-19 positive. No significant differences were observed in the sperm motility or morphology between the two groups. However, azoospermia was detected in 4 SARS-CoV-2 positive males. Since prior spermogram data were lacking, it could not be certain whether azoospermia was due to COVID-19. Although some studies support the negative impact of COVID-19 on male reproduction, they are limited by small sample sizes. Therefore, longitudinal study with extensive sample size in pre and post COVID data may better assess the impact of the disease on sperm parameter.